Hello again. In the previous video we mentioned different conjugation methods, and the first one that we introduced was the reaction between a primary amine and a carboxylic acid, which was called the peptidic bond or the amide bond. Well, if you know from the biology, you know that we have 21 different amino acids, and all the amino acids have one part with the amine group and another part with the carboxylic acid. If we connect different amino acids together, we will have a sequence of different amino acids which are connected to each other. What we are going to do, we want to know how we can use the affinity between the amines and the carboxylic acids of different amino acids to connect them to each other. Well, let me just show you how it was explained by the professor, and as you see here, it was mentioned that we just have a solid here, and we connect one of the amino acids on its surface, and this amino acid is connected in a way that the primary amine is available for the reaction. We introduce another amino acid, this is the second one, which can be connected to the first one from the carboxylic acid end. But on the other hand, the primary amine group is also available, and it can react with the carboxylic acid of another amino acid which is present in our reaction. So we have to protect this primary amine group, and the protecting group that we can use is called the FMOC group and the BOC group, as you see here. Okay, there's no need to remember them exactly, but just know that there are the protecting group which protects the amine here, and when these two amino acids are connected to each other, we will use the protection agent to remove it, to wash it out, and then the amine group, the primary amine group, will be available for being connected to another amino acid that we introduce. So the first one is connected to the second one, and when we remove it, we can introduce the third amino acid. You know, the third one can be also protected by another protecting group. Then we will remove it and we will introduce the fourth one, so we can have a sequence of different amino acids that we want. But the problem is that what is the best sequence of the amino acids? In order to understand and to realize, to find the best sequence of the amino acids which can be useful for our, let's say, for our application, we use the method which is called the Phage Display. Well, before we go for the Phage Display, let me just tell you something. You know, the peptides can be used for increasing the affinity of the nanoparticles or something towards something else. Let me just explain this way. If you see this virus here and the peptides at its end, these peptides have the affinity towards something else, towards a target, okay? These can be really useful for providing the targeting ligands on the nanoparticles. So when the nanoparticles, for example, have the right sequence of the peptides on their surface, and if you consider it as the cancer cells, when the nanoparticles are passing, they will be connected towards the cancer cells. So these peptides are useful as the targeting ligands. So we want to know the right sequence of the peptides which are useful for our application, and in order to understand, we use a library of different viruses which have been genetically engineered, and they display different types of the peptidic sequences. We just introduce them into our sample. We have the cells, and I don't know, it can be anything that we want to, let's say, assess the affinity of this towards the cells. And some of the viruses which can connect in a good way towards the cells will bind, and then we will wash all the other viruses, and only these ones will be connected. Then by doing some DNA sequencing, we can understand the exact sequence of these peptides, and we can use it to understand the proper sequence of the amino acids which has the affinity towards this cell. So when we understand it, we will use this method that we introduced here, and we will simply synthesize the sequence of the amino acids, the sequence of the peptides that is useful for our application. So let me just give you a brief summary. Just consider that we have a nanoparticle. Let me just use the draw here. Okay, just consider that we have a nanoparticle here, and this is the cancer cell. If we introduce some peptides on the surface of our nanoparticles that have affinity towards the receptors here, okay, the receptors which are here, these peptides can be used as the targeting ligands to connect to these receptors. It's like the attraction force between the nanoparticle and the cancer cells. So I told you how we can use this method to synthesize the right sequence of amino acids which is useful for increasing the affinity of this nanoparticle towards this receptor. And how can we realize what is this peptide, what is the sequence of the amino acids which has to be synthesized in order to have the attraction force towards these receptors. And we use the Phage Display method by genetically engineering the different viruses and to see which of them connect to the sample. And then by doing some DNA sequencing, we will understand the right sequence of the amino acids. Anyway, this was all about the macromolecules, and for the next videos we will go for the hydrogels.